Okay, moving on from chapter 8, where we talked about transcription, we are moving on to protein synthesis or translation to see what happens to those mRNA after they're transcribed from the genome. After 8 chapters, I don't think I have to say it anymore, but here is the chapter outline for your reference when you go back and study. So here's a 3D model of a ribosome from a bacteria. The ribosome is really important because this is the place where the protein products are made in the cell, and those protein products are what actually carry out function. So we did have a little caveat. We talked about those functional RNAs in the last chapter, but for the most part, proteins are what go around and actually carry out function. They're the enzymes, the transcription factors, the things that actually catalyze reactions and molecular pathways within the cell. And so this is a very important component of the cell, and because of this, it's also a big target for antibiotics in bacteria. And so more than half of antibiotics target bacterial ribosomes, which arrest this ability to create protein products, and therefore kind of it's either bacteriolytic, which is something that causes that bacteria to lyse and to explode, or bacteriostatic, which kind of stops the reproductive ability and kind of freezes the bacteria and doesn't allow it to continue to grow and cause infection. So now with modern technology, we can actually create these 3D models and predicted structures of ribosomes, but not only that, but we can also predict where antibiotic components and the development of antibiotics, where they will actually bind and target the ribosome. So the ribosome is a very important part medically of bacteria because that is where antibiotics generally, over half of them, as we said in the previous slide, have their function. But we're also going to go through this chapter and look at how they actually play a very important role in growth and development of cells. So in the last chapter, we talked about the classes of RNA. We had the functional and then the messenger RNA. The functional is the largest class, and within this class, we have two very important types of RNA. We have transfer RNA. It is this kind of adapter molecule that'll go and find the correct amino acid and bring it into the ribosome. We'll see how that works later. And then we also have the ribosomal RNA, or the rRNA, and this is a large complex that makes up the two subunits of the ribosome. And so these are both very important in translational processes, and thus we'll talk about them more in this chapter. So just like nucleotides and DNA, amino acids have kind of a general shape, a general formula, and have one little part that makes them different. And so in DNA, we saw those, the phosphate backbone, or the sugar backbone of the DNA, and then the actual nucleotides are what change. In amino acids, we have an amino group, which is the same, hence the name amino acid. We have a carboxyl group on the other side, and then we have this R group. This R side chain, the R in this case stands for the reactive group, and that's what specifies a leucine from a tyrosine or for some other amino acid. And so only the R group is what's different between these amino acids. So a polypeptide chain is a long chain or growing chain as it's going through the process of translation of amino acids. And so this continuous chain is added onto by the removal of this water group in between these two polypeptides or amino acids. And it's a bond between the carboxyl end of one amino acid and the amino group of the previous amino acid. Previously, amino acids were referred to as peptides. It's not really in vogue anymore. We generally call them amino acids, but they used to be called peptides, and that's why it's a polypeptide chain. A lot of peptides added together to create this long chain. So we really have 4 levels of protein structure when looking at amino acids and polypeptide chains. The first is the primary structure, and that is essentially just the sequence of peptides added to this chain. The second structure here is this initial local folding between polypeptides. And so you see that we start to get some hydrogen bonds that occur between different polypeptides, and this forms this kind of wavy 3D structure as opposed to just the amino acid chain. This also leads to the formation of things that we call beta sheets, and so we can get actual two rows of polypeptides that form these hydrogen bonds in between them and form kind of a flat sheet. You can get spirals, and you can get all sorts of different secondary structures because of these hydrogen bonds and interactions between amino acids in this growing chain. Thirdly, we have tertiary structure. This is where we have that long polypeptide chain. Even though it's formed these secondary structures like loops and coils, it is kind of the globular form of that entire polypeptide chain. So once you get the entire polypeptide chain all forming its secondary structures, that's kind of the tertiary structure, the globular structure. And then lastly, we have the quaternary structure of this. And the quaternary structure is generally, not all polypeptide chains have a quaternary structure. It's generally reserved for polypeptides that are part of a complex or are a subunit and require multiple subunits to form this larger complex. So in this case here, we have hemoglobin, and we have the 2 alpha and 2 beta subunits that make up this larger protein. So the beta would be the quaternary structure of this polypeptide chain. So one of the first big questions when looking at transcription to translation that was baffling scientists at the time was, is the DNA sequence overlapping or non-overlapping? That is, we could figure out the sequence of DNA. So here we have, or mRNA in this case, AUU, GCU, CAG, CUU, etc. down the line. Now, when this message was read at the ribosome for the creation of a protein, did it read 3 amino acids, then 3 amino acids, then 3 amino acids? We would call this the non-overlapping version. Or did it read 3 amino acids, then skipped 1 spot and read the next as 3? So we'd have AUU, UUG, then skip 1 spot and read the next 3, UGC, etc. And so what they found is that if you were to mutate one of these nucleotides, either upstream or within the mRNA, it caused a change in 1 amino acid. Now, if we were to mutate this amino acid in the overlapping model, we would have a change to 3 different amino acids, right? Because this U is here in the first amino acid, here in the second amino acid, and here in the third amino acid. So if we change that U to an adenine, so we had AUA, UAG, AGC, then we'd have 3 amino acids changed. So by looking at the mutation of a single amino acid, we could deduce that the way that this mRNA is read during the process of translation was to read 3 nucleotides, or as we call it, a codon, move to the next 3, move to the next 3, etc. Now, another question that arose during this process of unraveling how transcription and translation works was, how many nucleotides make up a codon? So remember, we have 4 nucleotides in DNA, and then 4 in mRNA, where uracil takes the place of thymine. So if we had 2 nucleotide combinations, so a codon was a doublet of nucleotides, we could have 16 possible combinations of those 4 letters, and thus 16 codons. However, that kind of brought up a problem because we know that there is larger than 20 amino acids present in most biological organisms. So the next hypothesis was, okay, what if there were 3 nucleotides used to create a codon? And in this case, we have 64 possible combinations of those 3 nucleotides, and thus 64 outcomes, or 64 codons. And so we knew that there had to be at least 3 nucleotides in a codon in order to properly distinguish between those roughly 20 amino acids. So how do we know that codons are units of 3? So we know it has to be at least 3 because we need at least 20 plus outcomes in these combinations of nucleotides so we can make the proper number of amino acids. So what they used are insertions and deletions in nucleotide sequences. So here we have a sequence TAGA-CAGA. So on the top we've inserted this T in between the A and the C. And at the bottom we have the same sequence TAG-ACAA. And there usually was this G there, but this has been deleted, right? So we have the same sequence, but we have one with an insertion and one with a deletion. And we can use this to deduce the number of nucleotides in a sequence, or in a codon, excuse me. So Crick was back at it. Crick and some of his associates took this FCO mutation in E. coli. So they have a gene that has a mutation that's indicated as green here. And he found that if he introduced a second mutation at some point, that mutated FCO gene would revert back to the wild type phenotype. However, while they were wild type, they were slightly different. So they weren't like 100% wild type. They restored that phenotype, but kind of like we talked about leaky mutations earlier in this quarter, it was a wild-type phenotype that was noticeably different than the non-mutated and rescued versions. So one of the classical ways that this is explained is by using three-letter words. And so we have the sentence here at the top, the fat cat ate the big rat. And so these are all three-letter words in a sentence. Now, suppose we have a deletion, which is a common mutation like that FCO example in the last slide. So we have the fat, and this used to be cat, but we have a deletion here. So now we have the fat ada, tet, heb, igger, at, right? So it causes what we call a frame shift. So the entire triplet of the that amino acid sequence is shifted off by one. And so that causes not only a mutation to this first amino acid that we had the deletion in, but every subsequent amino acid after that is changed because of that frame shift mutation. Now, say we had that same deleted C, that same sequence, and we add another secondary event, an insertion. And so this doesn't happen, so the C was typically right here, and so we're going to ignore the C. This happens 2 nucleotides down. And so it's not an insertion at the exact same spot, but it's relatively close. And what we see is that while this amino acid here that we have the deletion and the insertion in, that codon is still messed up, but every single codon after that has been rescued. And so the deletion by itself caused that frame shift, and then the insertion will rescue that original phenotype. And so, as you can see in the last example with Crick, we see that we pretty much, for the most part, have that wild type protein where we have out of the 1, 2, 3, 4, 5, 6, out of the 7 amino acids, only 1 is changed. So unless that amino acid is involved in binding, you're probably going to have very similar activity of this enzyme. So you'd say it's wild type, but maybe slightly changed. So now if we look at actual codons again, instead of the fat rat ate the lazy rat, or the whatever rat, lazy is not a 3-letter word. But anyways, if we look at a string of codons here that should code for amino acids. So if we have a wild type message, which is just CAU over and over again, and we have an addition of an amino acid, we see that we've got that frame shift again, right? So every single codon after that insertion is going to be shifted, and so we're going to have only one of all of these codons that had the original identity before that mutation. And so now, in the case with Crick's example, he had that original codon, and then they had that broken codon because there was an insertion, and then another codon down, the third codon, we have a deletion of what used to be an A inside here, and that restores the rest of the sequence beyond that. And so further experiments found that anytime you have a combination of 3 insertions, or 3 deletions, that we would have a wild type that was restored. So these codons, because it takes 3 always to restore the wild type, or 3 deletions to restore a wild type phenotype, it must mean that these codons are in triplets that read for an amino acid. What this finding of the triplet codon also revealed was that this code is degenerate, and that multiple codons code for a single amino acid. So we have phenylalanine has 2 codons, leucine here has 2 plus these 4, so it's got 6, etc. So if this wasn't the case, if we had a deletion or insertion, that could lead to smaller polypeptide chains, because it could be that we deleted and caused a frame shift that resulted in a codon being created that didn't have an associated amino acid. But even though you caused that frame shift and made all these new codons and mixtures of nucleotides, the polypeptide chain was still the same length, or in the case of a deletion, maybe 1 amino acid shorter, but not significantly shorter. So every possible combination of those amino acids, or sorry, of those nucleotides in the codon, leads to the depositing of a amino acid in this growing polypeptide chain. So every combination is covered by this code. So now we know that these triplet nucleotides are read in the process of translation, and that leads to the growing of this polypeptide chain. So let's talk about this process now. So we talked about the rRNA and tRNA earlier in this chapter. The tRNA, this is the structure of tRNA. It's kind of got this 3-leaf clover type look, an upside down 3-leaf clover from this point of view. But what we have is this anticodon loop. So mainly this is going to be what we're concerned about. So this anticodon loop for this tRNA has a complementary sequence that matches up with all of those different codons that we saw in that last table. And so for each codon in that last table, there is a tRNA molecule floating around looking to go and bind to that codon with its anticodon loop. And so here, this is just kind of the more 3D structure as opposed to 2D in the first example of one of these tRNA. So while tRNA is floating around in the cytoplasm, just hanging out, this enzyme, aminoacyl-tRNA synthase, will come around, and it is specific to each one of these tRNA. So in this case, we have the binding site for tRNA alanine, right? And so it will read this 3 codon, it'll bind to it, and then through this chemical reaction using ATP, it will bind that alanine to that tRNA. And then once it is bound, that aminoacyl-tRNA synthase can go and find another tRNA that is specific for alanine and continue to attach these alanines to that transfer RNA. It wouldn't be biology if there weren't exceptions to all of our rules for grading, correct? So we're going to talk a little bit about wobble bases here. So some tRNA can recognize 2 mRNA codons. It's because this third position is able to bind, so third position right here, is able to bind with either a C or a U. And remember, we had talked about how uracil replacing thymine means a bit more flexible with its binding partners and how it's able to form those structures. So the C and the U here are what we call wobble bases. And so this just means that for this AGG anticodon loop of this tRNA, it can recognize both UCC on the mRNA and it can recognize UCU. So just remember that it's not a 1-to-1 ratio all the time. We do have these exceptions called wobble bases where a single tRNA can read 2 different codons. So let's take a second to look at ribosomes and what they're consisting of. So we have prokaryotic and eukaryotic, and the ribosomes in these 2 groups are different. And so in prokaryotes, we have the 70S ribosome. 70S is the weight of the protein. So if you were to centrifuge it out or try to extract it, 70S is how we denounce the weight of it, so it weighs less than a eukaryotic ribosome. But it's made of two subunits. We have the 50S subunit, and we have the 30S subunit in prokaryotes. And within this 50S, we'll start on the left side here, we have a 23S RNA and a 5S RNA, and then 31 more proteins that make up that subunit. So this is obviously a protein complex that has not only ribosomal RNA, but also these protein subunits. Now, in the smaller fraction, this 30S ribosomal subunit, we have the 16S RNA, and this 16S rRNA is, you'll probably, if you do any work with prokaryotes and bacteria and genetics specifically of bacteria, the 16S component of the rRNA is something that's used a lot to develop phylogenies and determine what bacteria are related to one another, because it has it's kind of a sweet spot of conservation of RNA sequence where there's not a lot of mutations that occur in this 16S rRNA, but there's enough diversity between organisms that you can use it to distinguish from, say, a cyanobacter and a E. coli or a Pseudomonas or some other bacteria. So moving on to eukaryotes, we have an 80S ribosome, so it's a bit of a bigger protein fraction. And so we have a 60S ribosomal subunit and a 40S ribosomal subunit. And within the 60S, we have a 28S rRNA, we have a 5.8S rRNA, and a 5S rRNA. So you'll notice right away that this fraction subunit in eukaryotes is comprised of 3 RNA as opposed to only 2 in prokaryotes. And then lastly, we have this 40S ribosomal subunit for eukaryotes, and it contains an 18S RNA. And then both of these contain more proteins than their prokaryotic counterparts. So the 60S for eukaryotes has 49 proteins, and the 40S ribosomal subunit has 33 proteins. And so you can compare that to the 31 and 21 respectively for prokaryotes. So there are a few key sites of interaction in a ribosome where the ribosome is interacting with that tRNA and with the additional amino acid being added to the chain, the growing chain, etc. So we're going to point out a few of these key sites here. And so we start with the A site, which is down here. I hope you can see me circling it in purple there. The A site binds an incoming amino acid tRNA whose codon matches this mRNA. So this red line here is the mRNA. So this A site is kind of the first to read that codon, and it finds the matching tRNA, and we call it, in this case, an aminoacyl-tRNA because it's already binded with that aminoacyl synthase. And so it's got that amino acid attached to the tRNA when it's coming in. So it recognizes that codon, it finds the correct tRNA that has the amino acid attached to it, and starts the process of creating this amino acid chain. So next we have the P site, in that it binds the growing amino acid chain. So it's this amino acid, or this tRNA, moves over to the P site, and it's still connected to the amino acid, the tRNA, and it's just waiting for the next in line to enter the A site so they can kind of bind them together. So now the E site, at this point, the P site is still occupied by tRNA. The A site is accepting tRNA to add to the chain. Once they get to the E site, they kind of are pushing each other down this line. The tRNA is ejected, and its original amino acid that was attached to it is just left in that growing amino acid chain. We also have this decoding center. So the decoding center is kind of part of the A site down at the bottom here. And what it does is it ensures that the proper tRNA with the proper amino acid are being incorporated. If a tRNA comes in that does not match the codon that is required, then that decoding center kind of fact checks or checks to make sure that the proper one's there, and if it's not, it kicks it out to ensure that the proper tRNA can get in there and get in line. And then up at the top here, we have the peptidyltransferase center, and this is the region of the ribosome where the amino acid attached in the A slot and in the P slot, well, that reaction, that peptide bond that we had talked about earlier in this chapter, where that is formed, and those two peptides are linked in this growing polypeptide chain. If you remember the transcription chapter, we talked about how mRNA have this 5' UTR, which is the 5' untranslated region. And so the actual pieces of mRNA that are turned into the codons that create amino acids don't happen until you reach the start codon. So there are these codons and nucleotides upstream 5' of the start codon that aren't turned into protein. Well, within this is what we call the Shine-Dalgarno sequence. And so the Shine-Dalgarno sequence is this conserved sequence right here, AGG, AGGU. And that sequence is specifically preserved to allow translation of this mRNA to happen at the correct spot. So the 16S rRNA has a recognition site that says, okay, when we start reading this mRNA, we need to start at this Shine-Dalgarno sequence. And so that way it's positioned where the first codon is always AUG, as opposed to reading halfway down the mRNA or starting at the end. You need a way to recognize what the actual start of the mRNA is, and that's the role of this conserved region in the 5' UTR. Okay, so let's look at how translation initiation proceeds in prokaryotes. So we're going to start with this 30S subunit, and attached to this are two proteins, IF3 and IF1. And the role of IF3 and IF1 is to ensure that the 30S subunit doesn't go and bind to the 50S or to something else, to keep it free and open. Next, we have IF1 and IF2. And the roles of IF1 and IF2 are to ensure that only the initiator tRNA enters that P site, that first site in the transcription machinery or translation machinery. And so remember, the start codon to initiate the creating of a polypeptide chain is always AUG. So IF2 helps to recruit this initiator tRNA, and then IF1 helps to ensure that only that one gets into there. And at this point, the job of IF3 is done. So IF3, who was just making sure that the 30S ribosomal subunit wasn't bound to something else, now that that tRNA is there, it's kind of performing that function for it. It's in the way of it binding from something else, and so IF3 can leave. So now that we have that initial tRNA connected and bound to the mRNA, the 50S subunit, the cap of the ribosome, can come in and bind to the 30S subunit, the bottom, that was initially bound by those initiation factors to create or to prevent it from binding to anything else. So the cap can come in now that that first tRNA is there, and it causes those initiation factors to release. And so now we have a completed 70S initiation complex, the 70S subunit or unit of the RNA. So now let's talk a little bit about how translation is initiated in eukaryotes. So remember, we have some differences between the mRNA of eukaryotes and prokaryotes. First, we have this 5' cap, and that was that nucleotide that was altered to kind of distinguish the 5' end of this mRNA, and that helps to get the mRNA out of the nucleus and to the cytoplasm where translation happens. We also have, at the end here, it's not really indicated, but we have this poly-A tail, and that was to help protect it and to denote the 3' end of this mRNA. Well, within here, we have an AUG sequence. That's that start codon of this protein sequence. And we also have this 5' UTR that is the region upstream of that start codon that has that Shine-Dalgarno sequence and some other regulatory sequences. However, it does not get turned into a polypeptide itself. Now, the 40S subunit and initiation factors, similar to what we saw in the prokaryotes, will bind towards the start, the 5' cap of this mRNA, and it will remove any complex structures from the mRNA. Remember, as we showed previously, mRNA can form hairpin loops and all these different secondary structures because it's single-stranded and it's, it'll bind to itself in certain areas, etc. So we need to straighten that out if we were to read it and turn it into a protein. So this 40S subunit with those initiation factors bind towards the 5' end and help straighten out this piece of mRNA. Next, the 40S subunit will then start to kind of scan down that straightened mRNA, and it will look for that AUG start site. Now, once it's aligned, this methionine will be added as the AUG, the initial polypeptide of this growing polypeptide chain. And that 60S subunit then is allowed to come in and cap that ribosome. And so we end up with an ADS initiation complex that has the initial amino acid in place on that AUG site, the start codon, the first codon to be turned into a polypeptide, and we can proceed with translation of that mRNA. So let's add another layer of detail to this elongation process. And so when these tRNA are being incorporated into the ribosome, we have this protein called EFTU, so elongation factor TU. And what that does is it binds to the aminoacyl-tRNA and it helps it enter the A site, which this is kind of a bad picture because it's on the opposite side, but it comes around and it helps it to enter the A site. So once this tRNA enters the A site, the job of EFTU is over and EFTU leaves. At this point, because EF2 has left, it allows these two amino acids here to form a bond between each other. Now, once this bond is formed, there's another protein. This protein is called EFG, so elongation factor G, and what it does is it comes in and it barges its way into this A site. So remember, the A site is the spot where these tRNA initially will enter the ribosome, and it barges its way in. It's kind of a big bully of an enzyme, and it shoves everybody else down one more spot. So it moves the tRNA that was in the A site to the P site, and the tRNA that was in the P site to the E site, etc. So now this tRNA that is in the P site is kind of holding on to the chain while the next amino acid, in this case we have this purple amino acid, while it is entering the RNA with the help of EFTU. And as this occurs, this original blue tRNA, that has been barged down to the E site, the exit site of the RNA, and is allowed to eject out. And so remember, now that this tRNA is floating around out here in the cytoplasm, what's going to happen is that aminoacyl tRNA synthase is going to find this, and it's going to attach another piece of amino acid that matches its sequence, its codon, and allow it to be reused to continue to grow these polypeptide chains. So this cycle will continue until the ribosome reaches a stop codon. And so remember, there is no tRNA for stop codons. Instead, there's this protein called release factor that binds to the codon, causing the release of the polypeptide chain from the P site. So once we get to this UAA, the ends, the stop codon of that mRNA, this protein RF1, release factor 1, will recognize that stop codon, and there isn't a tRNA, but instead this protein, and it'll come in and shove the remainder of those tRNA down the line and cause the cleavage of that tRNA from that final, or that peptide from that final tRNA, and will kind of dissociate again the two subunits of RNA and allow this process to start over. So RF1 is responsible for coming in, knocking out the rest of those polypeptides, cleaving that chain, and allowing this process to start again. So in this translation chapter, I want to make a quick side note or continuation of our talk on alternative splicing. And so in our previous chapter in transcription, we talked about alternative splicing with RNA and how it could lead to striated tissue and smooth muscle tissue and brain tissue, etc. But usually what happens is we have very similar proteins that have small changes in them because of alternative splicing. And so in this example, we have a transmembrane protein that has this exterior domain. And within that exterior domain, we have different ligands that are able to bind to different molecules. So if we were to take just a snapshot of these two proteins that arose from alternative splicing, we see the orange proteins here, we see the fibroblast growth factor receptor. These are in the cytoplasm, so these are kind of the anchor proteins that are kind of holding these transmembrane proteins within the cell. And really the only two proteins that have changed are these purple proteins, which are our ligands, and then also the small under part here of these extracellular domains. And so a lot of times with alternative splicing, you have, because of this preserved amino acids for most of the structure, you have a very similar shape and function. However, you might just be distinguishing different targets because of this alternative splicing.